I may not be a rapper, but I am a scientist. And so are you, according to my analysis. Let's talk about CRISPR and how to change DNA, and hopefully you learn something new today. DNA is pretty sweet. It's what makes you, you, and me, me. But how does it work exactly? Let's think about DNA as the cookbook of life. It contains all of the instructions for making everything inside. DNA codes for RNA codes for protein. That's the central dogma of biology. Sometimes copying DNA, cells make a mistake. There are a whole lot of nucleotides, give them a break. And it turns out these mistakes are a positive. Over time, the diversity of life is what these small changes give. Some of these changes won't do much, just a small change in the function of a protein and such. But some changes will make that organism thrive, and some might not even allow it to survive. Once the mutations that help them live longer can reproduce, and their kids will have the same ones, which over time will diffuse. Generation after generation. More differences arise, that's speciation. We've been using evolution since the dawn of civilization, crossbreeding crops to improve them and feed a nation. When humans breed crops, it's artificial selection. When it happens on its own, it's called natural selection. Even with breeding, evolution is slow and hard to control. A lot of there is a faster way to get to our goal. Clustered regularly interspersed short palindromic repeat. Finding a good rhyme for that would be quite the feat. CRISPR is a bacterium's immune system. Cas is a CRISPR-associated protein. Consider that an axiom. Latching onto a particular gene is CRISPR-Cas9's talent. Scientists have figured out how to give it any given target. After binding to the target gene on another organism's DNA, what happens with CRISPR? You say, Cas cuts the targeted gene out, affecting protein expression, no doubt. It's like a mutation, but controlled in a lab, so we can create all sorts of changes. Pretty fab. But what if we could take this gene editing to a whole new level, without any genes being #canceled? Well, wait. There's more, because Cas9 just walked in the door.
Deactivated Cas9 confines genes just as well, but without deleting them. Now isn't that swell? Cas9 binds to a gene that was turned off, turning on that gene. What a great payoff!
Wondering what the point of all this experimentation was? There's practically no end to what gene editing does. Scientists can create crops more resistant to bugs, or even discover new drugs. One of the hallmarks of CRISPR's achievement, something really nice, was to engineer a crop now called golden rice. It has extra beta-carotene to produce vitamin A in populations that had a deficiency. CRISPR saves lives, but it's technology we have to be careful with, because its implications on our future are a monolith. CRISPR is changing our world, that much is incontestable. It's up to brilliant scientists like you to ensure its usage is always sensible.